We can see. Yeah, go ahead.
Great. Thanks. Hi, everyone. Welcome to my introduction to Biomedical Image Analysis Laboratory. I'm Vaanathi. So, before talking about my lab, I'd like to give a brief biography about myself. So, after completing my BTech in communication engineering, I switched to my research area and I started working in biomedical image analysis in my master's. And I was so fascinated by the subject and I never looked back. So, I did my master's in by research at IIT Madras in retinal image analysis, and I was working on identification of disease signs on retinal fundus images. And based on my work, I also have a patent on ophthalmic image analysis. So during my master's, I was working as research intern and project associate in Healthcare Technology Innovation Center at IIT Madras. So after finishing my master's, I got this exciting opportunity to pursue my PhD at University of Oxford with full scholarship. So I started working at Wellcome Center for Integrative Neuroimaging, working on biomedical imaging. So specifically on neuroimaging, again, identification of small vessel disease signs on brain MR images. So I won prestigious Prem Sukhi Foundation and Oxford Indira Gandhi Scholarship to pursue my DPhil. And I also did a brief industrial stint during my internship at Canon Medical Systems Europe Limited. So apart from brain MR imaging, I was also working on projects related to fetal ultrasound and chest X-ray images. So after completing my PhD, I continued as a postdoctoral researcher at University of Oxford. So basically, I was continuing my work on neuroimaging, but I was working on more advanced topics such as domain adaptation methods and also data analysis. So I currently hold honorary agreement with University of Oxford. So during my research career at Oxford, I have established collaborations with various groups, including the academic groups in the UK, USA, Italy, Denmark, and Iran. So after that, I got another exciting opportunity to work as a postdoctoral researcher at Harvard Medical School. So at I started working in Athinola Martineau Center for Biomedical Imaging, and after working on macroscopic imaging modalities at Oxford, I worked for first time in the microscopic data on dark field histological data along with MRI. So I have over 10 years of experience in working with medical image analysis and around 6 years in open source tool development for medical image analysis. So why work on medical image analysis, right? So currently the patient to clinician ratio in India is about 1,000:1. With such a patient burden on clinicians, automated methods for analyzing medical images could provide useful information to clinicians, especially radiologists, so that they can extract the information that's useful for the patient disease condition that could lead to early diagnosis of disease and timely treatment. So the main advantage of medical imaging procedures are most of them are non-invasive in nature, meaning that we don't have to do a surgical operation to or do any intervention to get the images. So we can most of them are like can be obtained without any surgery. So that is really useful in periodical screening. So that could again lead to early development and timely treatment, which increases the productivity of overall healthcare pipeline. So in this image, I have shown chronic health conditions that could be benefited from using AI methods or automated methods in healthcare. So first and foremost, diabetes with the incidence of 66%, followed by heart disease and cancer, 63% each, and neurological diseases. So when I say neurological diseases, it covers a wide umbrella of disease like neurodegeneration, like dementia, Alzheimer's disease, or cerebrovascular disease like stroke, which has high incidence in India. So all these diseases could highly benefit from implementing automated methods in healthcare and also infectious diseases. So how does the market for biomedical imaging look like? So the global medical imaging market has been increasing steadily, and it's still expected to increase by the compound annual growth rate of 5.8% from 2020 to 2028 and is expected to be valued at 56 around 57 billion US dollars by 2028. And if we look at the Asia Pacific market alone, the growth rate is going to be about 7.5 percentage. And you will also have wide range of job opportunities in industry, starting from medical physics in medical imaging, like the medical scanner vendors. The main players in this area are Philips, Siemens, GE Healthcare, Canon, Hitachi, to name a few. So apart from this, you also have wide variety of job opportunities in clinical and research applications in a wider range of industries. So ranging from computer-aided diagnostic solutions, pharma companies, genetic research, and medical instrumentation industry, as I was talking before. So here in these two pie charts, I have shown the most commonly used imaging methods, which is like CT and MRI dominate in the medical imaging industry, closely followed by X-rays and ultrasound. And with the recent advent of COVID-19, the X-ray imaging has proved to be even more useful. And looking at the specific organs of the body that has been analyzed more often, brain is more analyzed. So in recent years, people have shown interest in understanding the workings of the brain at both structural and functional level. Apart from that, again, as I said, the analysis of neurological disorders. And apart from brain, we have abdomen, cardiovascular, and lung imaging. So, what is biomedical image analysis? So let's get to the basics, right? So we have imaging data. It can be an MRI scan or a CT scan. So we have this imaging data. So our aim is to analyze this imaging data using a computational algorithm to get solutions that can give us useful information. So when we look at this image, this is one of the images from my study. So we see it as a brain, but how computer sees it is a matrix of multiple dimensions. A 2D image is a 2D matrix for the system, and a 3D image is a 3D matrix for the system. So I have just zoomed in to show that each pixel will have intensity values, and that is all the system sees the images. So it just sees it as a matrix. And when we perform different operations, so I have given you a few samples here. For example, if you enhance the image, what we see here is blood vessel and some organs here, but what the computer sees is basically the values in these areas. So if you increase the contrast intensity values, adjust the intensity values, you will be able to see the structures properly. So that's what we call as enhancing the image. And again, segmenting the structures of the image. So we know that this is a liver, this is an organ, some other organs in the CT image. But what the computer sees is this area is of interest. In this particular matrix, this area is of interest to the users. That's all the computer recognizes and sees. And again, registering the image, so transforming the matrices and aligning them on top of each other so that they can be um, easy to process. So we can actually, so what can you understand from here, right? So medical image analysis can be conceptually explained as an application of image processing and learning techniques on medical images or any images, but we are dealing with medical images for getting clinically useful information. So the structure here we segment, so that's clinically useful because the clinician can know where the liver is. So again an interdisciplinary field which combines the knowledge from various fields such as biology, physics, medicine, and engineering, because we need expertise on the anatomical structure of the structure of various organs to know what we are dealing with, and the disease progression to know what kind of signs we are pathologies we are looking for with related to a disease. So biomedical imaging can be done at multiple scales, multiple resolutions, and for multiple purposes. So it can, in general, meet two specific needs: clinical needs and science needs. So in clinical needs, our main concern is to get the results at the early stage, and also to get more accurate results by using an appropriate a medical imaging technique. So, for example, that's an X-ray image, CT image, and a brain MRI, magnetic resonance image. pancreatic ultrasound image, retinal fundus image, and positron emission tomography, PET of brain image. So these images could be used in a clinical setting to get to diagnose the disease at much earlier stage that could be used for timely treatment. Whereas for scientific needs, we are still at the research state where we are trying to understand what's exactly happening in the system. So even though macroscopic images are used more often in the science needs as well, but microscopic images are the most prominent imaging techniques used in the research setting, as in like for understanding cellular structure and functioning and physiology at cellular level for many organs. So the stages involved in medical image analysis, it's so we collect data from a clinical setting or a research setting for a specific study, and then the next stage is to process the data. So not all the data that's acquired can be quality. It can be of good quality. So we need to do a quality checkup and then do image enhancement or noise removal, and then that's the main part and the prominent area that we will be doing the research in BioMedIA Lab. So we'll be working on the image, medical image data, and developing image computing algorithms for getting clinically useful information. So these information can be for various applications. I'm going to talk about it in detail in upcoming slides. Um, but what is the specific research or the clinical problem, the healthcare problem that we are looking at, and what kind of useful information can we get from these images or the matrices as the system views them? So that's what we do in this stage. And next stage is validation, right? So we have developed a method, and how can we evaluate the method? So we have certain performance metrics like accuracy, how accurate the method is getting the specific structure, or classifying an image as a normal or abnormal image. And then finally deployment, right? So once we have validated the method, the tool can be released as an open source tool for deployment, or as a in a clinical setting, or as a software package in industry. So now we talk about applications of biomedical image analysis. The first and foremost, and probably the most commonly used application, is for diagnosis. So I have shown a brain image here and the lesions here. So these are the abnormalities, right? These are not supposed to be here, and these are one of the earliest signs for the onset of neurodegeneration, dementia, and stroke. So the first stage is to detect these signs, and the clinically useful information here is the actual detected areas and their volume. So we can actually monitor the progression of disease with the volume, and we can stage what we can decide what stage the disease in. And yeah, that leads to monitoring, another closely relevant area where we can actually study the progression of diseases. So we also have something called longitudinal analysis, where we acquire imaging data from the same subject, same patient in different time points, so that we can see how the disease is progressing at each time point. And then treatment. The main thing here is image guided surgeries. So this is also receiving very high consideration in the recent years, and this field is growing quickly, in a sense that so what happens in image guided surgeries is while doing surgeons, when they are doing surgeries, they get a real time feedback on the monitors in the areas that they are doing surgery on, so that they can do it with much more precision. So apart from these clinical applications, medical image analysis also has a widespread application in the research. So we need to, there is a lot we still don't know about biological systems. So for example, this is how a cancerous cell looks like as opposed to normal cell. So understanding this would help us in also studying clinical impact of various disease signs, and also various how various anatomical structures are affected by these pathologies. So another buzzword in medical image analysis is machine learning or deep learning. So I'm not going to go into much details here. I'm just going to give a brief overview for those who don't know what machine learning is. So it has three major aspects to it. So the first one is data. So we need to have data for training the system. And then the second aspect is feature extraction. where we extract useful information from data. And then the third aspect is training, where we train the model based on this information. So when we have this, when the data is acquired, this is the data that's acquired in a clinical setting. So we have this data, and how do we get these labels? So this is where, as I said, medical imaging is an interdisciplinary field. So we need a medical background to know what the pathology is and where the sign is. So someone who's an expert in biological systems, probably a clinician or radiologist would sit down and mark these labels. And then based on these, the data and labels will be provided to the system. And in machine learning, there is a step called feature extraction. So as Professor Atikanta was explaining before me, so images can be considered as a multidimensional tensors. So we can extract useful information like the value of features, the intensity values of various voxels, or the spatial location where exactly the coordinates. So these can be extracted as features, and then in an automated manner, of course, and then provided to the machine. Now the machine learns what the structure is and where the structure is, basically the labels of the voxels based on these features. And when the test data is provided, it provides the relevant output. So this is what happens in machine learning. And in the deep learning is very similar to machine learning, except for the fact that we don't have a separate feature extraction step, and we use more complex and deeper models than the conventional machine learning models in deep learning. So um, the image data is a multidimensional tensor or a matrix. So it has millions of voxel values to be determined, and therefore we end up with a lot of parameters. We are talking the order of millions for a deep learning model. So we need high performance computational systems, or the resources to run these deep learning models with the advent of graphical processing units and other high performance clusters. So it has been possible to apply deep learning models, especially in medical image analysis. um, and also, so in our lab, we'll be using mostly deep learning models for analyzing medical images. So another huge prerequisite for biomedical image analysis are imaging data. So there is another buzzword recently in medical image analysis called big data sets, because the larger the data set, the higher the statistical power of the results. You apply a method, you get results on the data. How can you say that this method is reliable? The more the amount of data, you can say with confidence that yes, this method is performing well on larger amount of data, so it's reliable. And also we get more data for training model. So having said that, small data sets have their own advantages. So even though they are not as preferable as big data sets, they also have their own advantages. So they represent the more realistic clinical scenario, because it's not always possible in a clinical setting to get large data sets. And also, small data sets obtained from multiple centers can help us to evaluate if our model is really generalizable, or if our model is able to adapt well to different data sets, because they are obtained with different scanners and they are going to have different characteristics because of different scanners and different parameters. So that's where the small data sets are really useful. So what do I mean by big data set, right? So typically imaging data sets will be in hundreds, more often in around 100 or under 200 subjects. Whereas in big data sets, we are looking at thousands of subjects. And one of the most popular large data set is UK Biobank data set. It is so far the largest brain imaging database. So they have currently scanned around 45,000 subjects, and their target is basically to scan 100,000 subjects. And the total database size so far is 500 TB. So that has multi-organ data and multiple scans like cardiac MRI and brain MRI imaging. And even within brain MR, they have multiple modalities. Um, and along with non-imaging metadata as well, for example, age, sex, and smoking status, other demographic details, and lifestyle factors, family, medical history, everything is available along with this imaging data, which is really useful for studying the clinical impact of certain diseases. We can study with respect to a specific population group. Right now, for getting access to this data set, application is required, and if the applicant meets the guidelines or requirements, um, so the data is available on stringent confidentiality agreement. So, what are the key research topics at BioMedIA Lab? So, we have a lot of exciting projects to work on. The first one is anomaly detection or computer-aided diagnosis, where the main focus of this will be to detect the disease signs or pathological signs related to the early stage of various diseases, and the main focus will be on neurological disorders. So, we will be focusing on brain MR images and other modalities to detect the early signs of dementia, for example, the small vessel disease signs. So, like for example, like microbleed, small blood leakages in brain, or small ischemic regions, and also the ones related to stroke. So, from the practical aspects of it, so we will also be working on projects related to domain adaptation or data harmonization to apply models on successfully on data from multiple centers or multiple sources, so that we know that the model is adaptable to multiple data sets, and also data harmonization where we harmonize data from different domains. When I say domains, it can be different scanner or different hospitals. So when we have these multi-domain data to more harmonize, so that the model can be used in a generalized manner for various applications. And then a semi-supervised and weakly supervised method. As I said, the main so in recent years, the main challenge in medical imaging is not the collection of data. So we have in the collection of large imaging studies has been tremendously increasing, but the main problem is to someone to sit and label the data, because that is labor intensive and expensive as well. So we need someone who has the expertise, for example clinicians, and we already saw that they have huge patient burden, so we can't get a lot of people to come and uh, mark or clinically or manually annotate the data. So how do we face this challenge? How do we address this problem of limited availability of ground truth? And also there are like a lot of inconsistencies in the ground truth as well. So how one clinical person views a sign and other clinical person view is are two different things. They have a lot of inconsistencies in how they mark the boundaries. So that is another important aspect that we'll be dealing with in our lab. So all of these are interconnected, right? So they are not separate aspects as well. So for example, we will be developing some method for anomaly detection where we get data from multiple centers and we don't have proper ground truth for the data. So these are some of the exciting projects to work on. So why join BioMedIA Lab? First is you will have access to UK Biobank dataset. And as I said, that's the largest imaging data, brain imaging data set. And you will have uh, da- data from multiple centers. So you can work on various pathological signs in both unsupervised and supervised manner. And apart from that, you also have publicly available data sets for this for various medical imaging applications. And you will also have access to high capacity GPU servers and workstations. And apart from this, you will also have access to shared computational facility from supercomputer research facility at IISc. And I have wide network of collaborators across the world on various ongoing projects. So you will have a research community to work with, and also for your further career options. And I am aiming to publish in high impact journals in the field, including medical image analysis, neuroimage, and IEEE TMI, and other top notch conferences like MICAI CVPR, ISBA as well. So the current challenges that I was explaining in the last slide, and also projects. So they are emerging as some of the hot topics in medical image analysis. So you will have wide range of exciting projects to work in. And if you have your own ideas, we can also manifest that. And if we can try to work on that as well. So you have exciting projects to work in, but not just limited to these areas. So we can work on diagnosis, monitoring, analysis of various imaging modalities with specific focus to the challenges presented in the clinical setting, like limited ground truth availability, small but data from multiple centers, and how do we deal with inconsistencies in the ground truth, and what if there are incidental findings wherein we have other pathological signs other than the ones we are focusing on. So how do we get the extra abnormalities? So what do we look for in prospective students? So good programming skills, of course, for in MATLAB or Python or C, and good academic track record. So innovative thinking when you are approaching a problem, and drive for research. So we are looking for candidates interested in machine learning, deep learning, pattern recognition, medical imaging, and computer vision. So the major interview topics will be signal processing, image processing, probability, and machine learning basics. So the candidate should definitely have some basic ideas in signal processing and machine learning basics. So yeah, so for further information, you can contact the CDS office, and also please refer to the admission brochure. So good luck with your interview preparation, and I'm happy to answer any questions if you have.
Yeah, thank you, Vaanathi. That was a very nice description of the lab. So, are there any questions?
Oh, I see something in chat.
Yes, some.
So what background for this lab should be sufficient? Yes, as I said, I'm looking for candidates having some background in signal processing, and some aspects of probability as well, because we'll be dealing with a lot of probabilistic models, and machine learning. Um, so that's what I'm looking for. And as I have a paper in the same domain, other than that, I have reviewed so two. What is SCI? Science paper, is it? In the
No,
same.
sir, some Scopus index, something like that.
I
So,
see.
Vaanathi, can you stop sharing your screen? I think.
Oh, sorry.
Okay.
All right. Okay.
Okay, good. So yeah, so Karan, the thing is that the interviews will be mostly on the basic subjects, right? So linear algebra, probability, things like that. Once you have those in place, right, as we have been saying so far, that is sufficient, right? So your specifically what your background degree is not important in some sense, right? So it is what you're interested in doing, what skills you have. That is what will be judged in the interview. Okay. Yeah. So.
Okay. It means this lab is only to deal with images. For now, yes. So in future, we are also planning to include NLP, natural language processing, to get radiological reports from imaging. But in one way or another, we will be dealing with images. As the lab name suggests, the main focus will be on image analysis. So when we look, sorry, Deepak, did you have anything to add on this?
No, that's good. So the other is the time series analysis. Are you also doing time series analysis? And
So that's very close to longitudinal image analysis, right? But using imaging data rather than the signal. For example, if you are looking for EEG or ECG signal, so that's not currently the focus of the lab. But if there are any very interesting projects you want to work on, you can always email me and we can see how to take it forward. Um, can you explain something about future scope? So the future scope is mainly moving towards how to explore the pathological science. As I said, the earlier the science, the better. So rather than the very advanced science, people are looking at early stage science And the main problem with that is that they are quite small, right? So you don't have very clear ground truth. So the future is moving towards how to address the challenges in medical imaging by getting uh, the small data without ground truth, or even like how to adapt in real time, like test time domain adaptation or radio mix. How to, if we get a sign, how, what can we say about the sign, like which is clinically useful for the radiologist. For example, they actually say that this is the one, this much quantity is there in this location. So how can it make it of real use to the clinicians? So those are some things that are of future scope. um,
Yeah, Yasir, you had a question. You can unmute yourself and speak, Yasir.
First of all, good evening, madam, for presenting such a good presentation. And I have a question that in this field of in this lab, uh, actually, I have my means actually consequently I have also my MTech thesis work in medical image processing.
okay.
In this lab, what are the scopes we of doing a student who is belong to medical image processing background? What are the scopes we can get in this lab? Actually?
So you are asking if there is
What are the
project.
opportunities?
Sorry,
What are the opportunities are there?
for non-medical imaging. Is that what you
No,
are?
no, no, no. For medical image processing background students.
Yes. So you can. So I have. We can work on projects related to computer-aided diagnostic and the diagnosis, and then modeling specific structures in the image. Basically to understand. Um, the clinical impact. So it depends on what kind of specific aspect you want to work on. At subject level, for an individual patient, you can see how healthy he or she is. So by getting how normal the anatomy of the organ is, if you are looking at brain, how normal the brain is, and what kind of abnormalities are present in the brain, and what could these abnormalities be due to. If we are looking at population level, we can see how this disease is affecting that whole population by modeling
Yes, ma'am.
the progression of disease. So we have different an appropriate project if you want to work.
Ma'am, actually, in the middle, you mute yourself.
Did
No,
I?
no, Shah. Yasir, what happened is some train went behind you, and that's why that happened. So,
Okay, okay.
okay. So, Vaanathi, I think the last last 30 seconds or so, we didn't hear because of that train. So,
Oh,
can you please repeat?
yeah,
Yeah.
I was just saying that it depends on whether you want to do it at subject level or at population level, whether you want to do the detection of disease sign or model the progression of disease. We have relevant projects, and you can choose what is more interesting to you, and we can start working on it. Yeah, so that's what I was.
And madam, what types of images basically currently the scope in research? Means it is a CT scan, MRI, or echo.
I mean, all of them, right? So if you are looking at a low, so MRI and CT would require high-end scanners, whereas ultrasound and X-ray are quite easy to acquire. So if you are looking at something to work on at at a low end, so you can work on X-rays and ultrasound as well. If you are looking for identifying disease for research problems using MRI and CT, so that's also possible. So it depends on what kind of clinical setting that you are working in.
Yes, ma'am. Ma'am, in the slide
So,
you show one thing.
yes, yes, as it we have run out of time. You
Okay,
can
okay.
please email Professor Vaanathi. In the brochure, her Google Scholar link is available, so you can go there and find her homepage and email her. Right. So then
Yes, yes,
she
sir.
will
Yes, sir.
respond to you further queries. So we'll stop today, right? It's been a 2.5 hour session, and everybody is probably very tired with that. Once again, thanks, Vaanathi. So actually, it's good morning for Vaanathi. She's currently in Boston, right? So thanks, Vaanathi, for joining early in the day and talking to our prospective
No,
students.
thanks. Thanks, Deepak. It's my pleasure. So I hope you
Yeah.
all got an overview of the lab, and
Yeah.
looking forward to seeing you all in the interview. Thank you so much, and good luck with the interview process.
Yeah, good luck. So, thank you. So, I will be closing the meeting now, and tomorrow you have the sessions by the other EECS labs. Okay, so students, you can attend tomorrow's sessions. So, the CSL lab, Dream lab, VAL, VCL, Mars labs, right? So, they will be talking tomorrow. Okay, so take care then.
Sir,
Bye.
sir,
Yes.
sir, I have a journal question I want to ask about you.
Okay.
In the email in which I got call letter, there were two schedules. One was for interdisciplinary program, brain and artificial intelligence, right? That is on 21st. It is associated with you. I will be giving a test on 10, or I'm not getting any information regarding that.
I didn't understand.
Sir, you are organizing the test on 10. Yes, sir.
CDS is a different department. Brain and artificial intelligence is a different department, right? So everything is different. So everybody will have their own interview process. So you have to contact the brain and artificial intelligence department for their process. So we are here only for CDS, right? So computational data science department. Our process is what I am describing, not any other departments.
Okay, sir. I want to know if there is any method to contact. I'm actually trying to call them for 2 days. No one was answering, and
So there
uh,
must be a admissions contact on the call letter. You can contact them.
okay, okay. Thank you, sir.
Okay, so take care then. Bye.